Okie dokie, everybody. We are going to be going into the fifth section of our notes, which is going to be about the structure and function of biological macromolecules. Didn't we already learn about the structure? We are, but we're now going to understand a little bit more about how these structures actually form or why they're formed. You want to pause the video, you can go and take a look at this. But what this is saying is that the directionality of the subcomponents will influence the structure and function of the polymer. Guys, it's nothing different we've been talking about, right? The order in which things are going to be presented within our molecules will determine what its properties will go ahead and be. If I have a set of instructions that tells you how to make a peanut butter and jelly sandwich, you'd imagine it'd be get the bread, then get your peanut butter smeared on one side, get another piece of bread, do the jelly, right? And you make a sandwich. But if I tell you to get the peanut butter first, and then you get the bread, and then you get the other piece of bread, and then you get the jelly, you can kind of see how the instructions kind of don't really line up or work as well, right? That's basically what this is referring to. So we're going to go ahead and hit all these things, specifically making you aware of this 5' to 3' regions again, as well as talking about how protein specifically fold, because protein folding is a really fascinating subject. All right, so this is a nice little tiny review chart, I would say, that I would maybe ask that you copy or fill in, just so that way you can kind of keep track of all the awesome things that we're taking a look at here, okay? So if we go and take a look, I'm just going to kind of like get some of the words out of the way, right? So we can see we got our monomers, our dimers, and our polymers. So we already know that monomers, like those individual units, while the polymers are like being combined into larger and larger. A dimer is just a two section, right? So in the case here, that's going to be like a disaccharide. Example of a disaccharide would be like sucrose, which is made up of a glucose molecule and a fructose. They come together. You also have things like lactose, which is galactose and glucose that are combined together, right? So, there's a couple of examples of some disaccharides that we end up having that are in there. When it comes down to the polymers, right, again, we have your other names. You have like ones for plants, you have two for animals. For humans or animals, glycogen is how we end up storing our energy in long sources, right? That's all carbs. In case of proteins, right, how many amino acids that you have, you can go ahead and fill that out, right, from the last video. Any examples, you just go to that chart, right? When you end up putting two amino acids together, you get a dipeptide. And then the general name, right, we're going to go and show you all this stuff right here. We're going to go show you what does it mean by a primary structure and a tertiary structure. That shouldn't make any sense right now. We haven't talked about it. But basically, it's how you get this whole line of amino acids and how you make these like really weird funky shapes, with the ribbons and stuff. You've probably seen this before, maybe, and you're like, I have no idea what I'm looking at. That's perfectly fine. We're going to go and actually get you aware of it. And finally, you got with your nucleic acids are made of your nucleotides, right? And we know of our different types of nucleotides. And there's no dimer. There's no two nucleotide sequence thing that ever exists. That's not a thing, right? We just have nucleic acids. They come together and we make DNA and RNA with them. I'll explain why they are structured the way that they are. So we're going to start with nucleic acids. They're relatively I'd almost like argue they're like the middle ground, kind of a little bit difficult, but they're not that bad. So let's talk a little bit about these guys, right? So last video, I ended up saying that the difference between ribose and deoxyribose is this little spot right here, right? Now, ribose sugar will have an OH group, while deoxyribose means, remove oxygen. Deoxyribose, right? Oxygen. We literally just got rid of that oxygen that was on that second carbon right there. That's it. That is the big difference between those two pentose sugars. Right? That's one structural difference that's there. Now we end up saying that they have distinct ends as 5' to 3'. I was mentioning it when we are building our nucleic acid sequences, right, or DNA and RNA. There is going to be the 1, 2, 3, 4, and 5, the 5 positions of where these carbons are, and we build it 3' and 5'. Well, it's specifically 5' to 3' is the actual directionality of it. So we're able to see that, okay, cool, like we're attaching the 5' of this region, sorry, I should say the 5' of this region to the 3' of the next one, right? So see the 3 here, 5 right here, 3 right here, and that's how it ends up combining. Okay, it's all going to be covalent bonds, right? Dehydration synthesis, a condensation reaction, dehydration synthesis reactions in order to go and build these guys, right? Some things to kind of keep in mind is that we have to go look at the directionality. I was talking to Miss Tran, and she uses a cupcake example, right? Just notice what direction that the pentose sugar is moving. It's going to be pointed up. It moves in that direction. And when we go and take a look at the opposite side, which I'll show you in a little bit, it's going to be pointed downwards. It's going to be like going like that direction. We'll show you why in a little bit. Okay. So it says two strands are anti-parallel and they form a double helix. What the heck is anti-parallel, right? Well, if you think about lines being parallel, this is an example of what a parallel line will look like, right? Now, you're probably thinking anti-parallel, that sounds like it's perpendicular or something. That's not the case, right? In the case of it being anti-parallel, it just basically means that one side is going in one direction while the other one is going in the opposite direction. And that's what I mean by they being anti-parallel. So what we're going to go see is that the DNA is going to be almost like facing up in this direction and facing down in that one. We'll go and see that. Now it says that the direction and the number of hydrogen bonds, right? We had to go take a look at that. All right, let's go take a look. Notice how this guy is facing up. Notice how this guy is also facing up, right? And again, we have our 5 to 3 sides. Or 5 to 3, right? We're attaching ourselves 5 to 3 prime. If we go and take a look over to here, I'll do it in different colors so you can actually kind of see what's happening. You get like a purple, it'd be fun. Notice how the pentose sugar is like upside down now. It's upside down. Notice, right? So we can keep on saying, right? So if we're taking a look, here's our 3, here's our 5, here's our 3, here's our 5, right? Et cetera, et cetera, et cetera. Now, what happens is that this is how DNA needs to go and be done. The reasoning behind this is actually it actually has to do with these guys right here, specifically the hydrogen bonds, right? So the base pair rule, if you know, a Char-Gas rule, it's like A pairs with T, right? And then G's will pair up with C's. In the case of when you have RNA, right, A is going to be paired with U, right? I'll just do a little star right there just so we get that. But what we're getting at is that, okay, so the thymine right here is going to be attached to the adenine, which is what we see right here. Okay, and then I'm going to go ahead and do the yellow right in here for this one, right? And then the guanine and the cytosine, right? That's this combination right here. Now, you're probably wondering, wait, why is this specifically relevant? Well, what happens is that we can actually form hydrogen bonds between these elements, sorry, these nitrogenous bases right here. In the case of guanine and cytosine, you're going to go and make 3 hydrogen bonds. And in the case of our the adenine and the guanine, right? A and T, right? You're going to get 2 hydrogen bonds. We can count them right here. Here's 1, 2. A way to kind of remember which one is which is I always like to do this. I always think of the what is that? The all-terrain assault troop carrier. I don't know, the AT-AT from Star Wars. Can't remember what the thing is. It's a big giant like landwalker thing. You may remember. It's like the guy looks like it has like a cow head with little lasers. You go pew pew pew, right? That thing from Star Wars. If you haven't seen it, you got to go watch it. Anyway, this is my remembering that this is the one that ends up making the 2 hydrogen bonds. While in the case of, this guy over here, the GC, I would say GNC or GNC, right? Kind of like the vitamin company, right? There's like 3 letters, 1, 2, 3. That's how I kind of remember which one's which. Do you need to be aware of it? It just shows up in there as the things you got to be aware. So these little dumb tricks are the things that help me out for remembering which one's which. And I'm probably wondering, like, well, why is it that it works this way? Well, let's go and clear this off for a second, okay? Let's say that if we had a 5' to 3' over here, right? Just how you see it over there, and you have a 5' to 3 prime over here, right? So like the exact same idea. And let's just say hypothetically they were lined up, right? So I have a thymine over here, I have the adenine up here, I have the guanine down here, I have the cytosine, right? So just imagine that that's it, but we're still like going, we're parallel, not anti-parallel. Well, the problem ends up being is that how this shapes, if you imagine like flipping it, right? So if we were to do like a 180-degree flip, right? A 180-degree flip, we wouldn't be able to make these hydrogen bonds that are right here. Like this would not be able to happen. They wouldn't line up correctly because if you think about it, right, this oxygen is negative, right, and we have some positive regions over here, right, with the hydrogen bonding. But if we flip this over, there's nothing for this oxygen to grab onto, so it doesn't actually bond to it, right? And it's even more pronounced if we go and take a look at guanine and cytosine. Like this one is like, I think is when it ends up, getting a little bit more particular about it. Okay, look, right? Remember like how like oxygen is negative, right? So this oxygen is negative over here. Okay, cool, really awesome. We'll flip it over so now both the oxygens are facing each other. What do you know it's going to happen to those intermolecular forces? Are they going to be attracted? Heck no, man. They're going to be repelled, right? So then you can't even form hydrogen bonds right there. So again, like the reason why it's anti-parallel literally is so that the hydrogen bonds are actually able to form correctly. It's kind of a wild thing, but I always like to kind of like, bring people's attention to that. All right, quick little thing that's just kind of showing to finish up with our DNA, right? They are anti-parallel, right? We can see our 5' N going in this direction, right, to the 3', and we have 5' N going down this way to the 3', right? We can see this, right? We know that the hydrogen bonds are forming up because, right, because that there is there's spaces for them to go and interact with, right? This is negative, that's positive over to here. This is going to be slightly negative, and this one's going to be slightly positive. We go take a look here. This is negative. That's slightly positive, right? You could go down on the whole entire way and be able to go and see that stuff, right? And that's why it's set up the way that it ends up doing. We end up calling this. Okay, we haven't really talked much about DNA, but this is called the sugar phosphate backbone. Phosphate backbone. And this just ends up describing, the rungs of the ladder, right? If we have a ladder, here are the rungs, right? They're the opposite sides of it. And then if I'm looking at like my hydrogen bonds, right? The hydrogen bond, this is like the rungs of the ladder that we can see right here, right? So it looks like this, right? This is where your hydrogen bonds are, and this is how we actually have that DNA set up. Now, it's actually a twisted double helix because there not only do you have like these things happening, right, but because these phosphate groups and they're all like negative and stuff, and all these negatives are happening, there's going to be positive regions. So it kind of folds in on itself because there's going to be hydrogen bonding like due to the spaces in here. We're not going to go into detail on like more about it, but just know like hydrogen bonds like show up freaking everywhere. Like they are the best intermolecular force in order to go and hold all your biological molecules together. All right, now we're going to go ahead and get into the realm of amino acids. Okay, so if we go and take a look at this image, we'll see that we're just combining amino acids through dehydration synthesis and creating peptide bonds in order to go make an amino acid sequence. All right, let's actually kind of break this down because this is kind of really important for you to understand, right? When it comes down to proteins, they have four structures. They have what's called a primary structure. They have a secondary structure, or secondary structures, I should say, secondary structures, tertiary, and finally quaternary. Now, quaternary. Just to give you a heads up, all right, quaternary is a little bit weird. It's kind of like the pseudo one on top of them, which we'll show you momentarily. But like these guys, it's really really neat, and that's what we're going to go highlight now with proteins and why proteins fold the way they do. Okay, now we are not doing any protein folding. This primary structure is the sequence of amino acids. There is no folding. We are not folding. All right, we're not folding our amino acids. All we're doing is just we're saying what is the order of stuff. And it's like, okay, cool. You got, I don't know, glutamic acid here. You got thiamine here. You got alanine here. You got tryptophan right here, right? We go on, go on, go on, right? This is that amino acid sequence where you would get like your codons, your TTC. You'd then transcribe it into the AAG, and then you'd use the codon, the codon chart or whatever, and you'd get whatever amino acid you'd end up getting. That's literally all this is. Like that's all that is. So this process right here is just talking about your amino acid sequence. Do we need to know about the order? Well, it kind of does make a difference, right? Because if you remember, right, some of these guys might have like negative charges associated with them. Some of them might end up having, oh, I don't know, like the positive charges associated with it. So you got like a positive right here, right? So then this positive is going to be interested in like hanging out with a negative somewhere. Maybe you have like a hydrophobic over here, hydrophobic region. So we're going to go play around with protein folding in a classroom. It's a little bit of a goofy activity, but I think you'll end up having fun with it and kind of understand what's happening there. All right. That's all the primary structures. It's just the order in which the stuff shows up. Nothing really interesting. Secondary structures. Okay, this is when we're starting. We are starting to fold, right? We are starting to fold. Starting to fold. Okay. Now the secondary structures are like the most common. Like it's the most common stuff that you see. Okay, and there are two different structures. You have the alpha helix and you have what's called beta plated sheets. Now, if you like just Google search a protein, right? Like Google search protein, like do this right now. Like I'm even kidding, like protein computer model. All right, and I want you to go and see if something shows up. Okay, and hopefully just do that protein computer model. You should see some weird funky ribbon shape where it's like it looks like some ribbons, right? You might end up seeing like some weird stuff, and there's like other ribbons, and it's like twisting around and more ribbons and stuff like that. And you're like, I don't know what I'm looking at. Well, those ribbons right here that you see in these images, that's these alpha helixes right there. So it's actually just how the protein is folding. And then if you see it almost like going like back and forth, like almost like a swishy kind of thing, right? Like this swishy thing right here, that's going to be the beta plated sheets right there. And all they are, it's how the protein is going to fold, and it's just due to hydrogen bonds. Like they both are just hydrogen bonds. It's just how is it that the protein is going to go and fold, and that's all it is. It's just ideally like making these helixes, right? Similar to how like DNA is. Again, this is not DNA, right? But it's like the same principles are happening because DNA is like a twisted ladder. All it is just these hydrogen bonds that are just holding it all together, and it's just really, really freaking easy for us to make all these hydrogen bonds. Like, look at them; they're everywhere, right? And this is just because that's how the amino acid goes, right? Now, where it gets a little bit more crazy. I'm telling you, like this is cool. Like I this is where I start to love proteins. Is where I start to talk about tertiary structures, right? Tertiaries are like the big 3D curves and folds. Like this is when you're doing like origami, right? And like how like you do like the crazy like paper goes all the way over to here. Well, the tertiary structure is like the final way. It's like the final fold of the amino acid sequence. And the reason why I say that is that there's no more folding on top of this, right? Like we're going to go and fold. We're going to go make hydrophobic interactions, right? So you go take a look. This is hydrophobic. This hates water. Hates water. Right? It hates water. So it's going to fold in a way so that all those hydrophobic areas are just nowhere near each other, right? We can have having an ionic bond because we have a positive and a negative region close with each other. Like, boom, we're going to go and get those guys attached to each other. We can use sulfur to make disulfide bridges where just sulfurs come together and it just happens. And then we can finish it up with some more hydrogen bonds. Like, we made our helixes, we made our beta sheets, but now we're going to go ahead and do our hydrogen bond. And literally that is like how proteins fold. They're probably always like, wait a second, Masaki, you said quaternary. Where are the quaternary structures? You said to write this down. So again, this is a final fold, okay, of like one amino acid sequence, right? Okay, this is one amino acid sequence. When you go and take a look at these ones, the quaternary structures, we're basically making, three or four different amino acid sequence come together, right? We can see, hey, there's those beta sheets, right? There's those alpha helixes, right? They're right there. You can see them all over in these guys. And what happens is that there's going to be some kind of folding with themselves. Like they fold into each other. It's like you and I are going to go and build a car, right? Like you are building a car and we're building a car together. And you end up coming up here with the engine. And then I come in here with like the tires, right? And then someone else gets the alternator and what the heck ever goes into a car to make it work. But then at the end of the day, like this whole thing is the car. This whole thing is the protein. Like this is the protein that does its job. And what's neat is that they are designed to fit with each other, or to like just do a job, right? Remember, I keep on saying, right? The structure, the order in which these subcomponents show up determine what it's capable of doing. For example, you got these enzymes, right? You have like lactase, which we'll be playing around with in class. Lactase breaks down lactose. It's specifically a milk sugar, right? Well, you also have what's called sucrase. And sucrase is designed to dissolve and break down sucrose. Well, sucrase is great on sucrose. It wants to be used on sucrose. But if I end up using this on lactose, like milk sugar, this is not going to work because the structure of this amino acid sequence is different from the structure in that amino acid sequence. Even though they both break down sugars, because they're slightly different because of how these ribbons and disulfide bridges and subunits, all these other things are happening, that makes it so the protein actually has a specific function. And what's really cool is you take it a step further and we say like, okay, well, let's go ahead and adds hydrogen ions, right? It's like, oh, cool. Now we're in an acidic environment. Well, keep in mind, right? Remember like how you have those hydrophilic interactions, or you have those ions. Well, they might end up starting to being attracted to this, causing this to unfold, and then the protein just won't be able to fold itself back in. And this is when we talk about denaturization. I'm kind of getting on a bit of a tangent, but like, and we're going to get to that later on, but there's so much stuff that is happening, or so that can happen to this. So protein folding, I think like this is like, I call it like the chef's kiss of biochemistry, because if you can understand it, like you're going to be good for understanding anything that we're going to go hit in this class. You'll understand mutations, you'll understand chemicals that affect the proteins and all that other jazz. So let's actually like look at this, right? So here's our denaturization right here. We're just taking this protein, we are going to go and do something to it, whether it's like heating it up or exposing it to salt or exposing it to pH, anything, like any of these things, like it would be bad. These proteins are designed to be at a specific pH and a specific salt concentration, and if you end up changing it up, they just stop working, right? Like your stomach has an acidic environment for your digestive enzymes. Well, once you end up like taking like Tums or something and slightly changing the pH, and you make the pH end up going higher, well, now those enzymes aren't working as much, and it kind of helps out for the acid reflux, but now you're not getting as much digestion going on in there. Like there's so much little things that are happening within there. It is wild. Okay, the last one. Hey, this should look a little bit familiar because we talked about this in the banana lab, right? Starch and glycogen and stuff, right? Carbohydrates form linear chains of monosaccharides, and it can be linear or it can be branched. I did not end up going into showing you a picture of what cellulose looks like, or what chitin looks like, but basically, like it's just like cellulose looks like this. It's like the linkage, or the glucose linkages look something like this. And if you kind of think about this, right, if I were to put like a little box right here and a box right there, you can kind of start to see like how that might end up looking like the leaf of a plant, right? And that's what they're made out of, right? Cellulose. In the case of us, we have glycogen. This is in your liver, right? As you keep on saying, this is used in order to break down, and we turn it into little glucose molecules. So in the event that your blood sugar, right, if your blood sugar is low, your blood sugar is low, your body will end up going to the glycogen in your liver, breaking it down. So then that way we'll go and end up increasing the blood sugar with putting glucose in your body, and then you end up, increasing your blood sugar. Chitin right here, that is going to be found specifically like in exoskeletons. If you think about crabs or lobsters, or if you think about insects, they have like a crunchy outside that's made out of chitin that's primarily made out of those carbohydrate sequences. Another really cool thing about this, and it's just one more thing to leave us on, and then we'll be done with the video, is a way on how we use these resources to either uh, grow or get bigger. And here's what I mean by that, right? If you go and look in the mirror right now, right? You're standing in front of a mirror, right? And the mirror is just like, hey, look, I see myself in here, and what am I made out of? Right? And you say, like, what am I made out of? Well, you're primarily made out of proteins, right? We showed you this before, right? You're primarily made out of protein. But if like a plant would end up, putting itself like in front of a mirror, right? And we put like the flower in front of here. Well, the flour is not all made of proteins; it's made more of carbohydrates. And what's really neat about this is why. Well, hydrate. The reason why behind this is that, well, how do plants primarily get their nutrients and their energy from? Photosynthesis, right? So, and when we talk about photosynthesis, they're constantly making glucose. They're constantly making glucose. So they're literally building themselves out of carbon dioxide and glucose, and that's how they make their starch. And that's also how they end up making their cellulose. Versus us, well, we don't do photosynthesis. Instead, we eat, we consume. And you end up eating the carbs, but you're primarily eating those proteins, because those proteins are what makes you be able to go do all the crazy fun and awesome stuff that you do. So it's just really cool. It's really interesting how depending on your method of energy and nutrients will really depend on how you end up the organism ends up building up. So uh, because of the interest of time, I would rather just go ahead and go through nucleic acids as well. All right, so nucleic acids, because it's going to go into more detail. Forget about it. We'll do it later on. It's fine. If you want to go take a look at it, you can. It's literally going to be a quick guy, everyone. It's like we're just going to talk about the differences between nucleic acids, DNA, and RNA. It's going to be a quick guy. We'll get into another time. All right, that'll be done with this video. Have fun. Come with your questions. If you have any issues, let me know, and we'll have a good time, and we'll hit this last guy next week. Take care.